Good day. In today's lecture, we will be discussing about the archaea, the archaeal cell structure. So the reference for this lecture would be on Prescott's Microbiology 12th edition. So in this part of the lecture, we will first talk about what is an archaeal cell. So let's look at the typical archaeal cells and how are archaeal cells different from what we know of bacterial cells. So, Archaea is a relatively new study or a newly discovered, it's relatively new. Uh, Rather, scientists realized that we have fairly recently that we have a very separate group of organisms called the Archaea that is different from bacteria. Before, what we know of the Archaea are part of the bacteria, or rather, we do not really know that there is a difference or a separation between the two. So archaea and bacteria were all of them were classified originally as a one group, and there we call them the prokaryotes. Now, because of the progress in the technology and with what we know of molecular biology, we now realize that the archaea are actually a distinct class of organism with very different characteristics from the bacteria. Because remember, previously, bacteria and archaea, since they are all single-celled organisms, we can only know of their existence, or rather we can study them mainly through the use of microscopes. And even then, it's very difficult to distinguish one from the other. In fact, before the advent of the DNA technologies, some archaeas are named and classified and identified even as a bacteria, or rather within a single bacterial species, but instead of being their own taxonomic class. So, archaea are highly diverse with their morphology, physiology, reproduction, and ecology, but we only now realized that that a distinction, although we are aware of that there are some bacteria that does this and that, before we classify them as bacteria, but now these are distinctive taxonomic taxa. They have their own morphology, physiology, reproduction, and ecology that is vastly different from bacteria. So, archaea are best known for growing in anaerobic hypersaline conditions. When we say hypersaline, they are very, very salty with a very, very high salt concentrations. On extreme pH, so it's either very acidic or very basic, and even high temperature habitats. So even in, basically, we usually see them in volcanic vents, in the underwater volcanic vents. Those are very high temperature, of course, because that is where the lava flows. And of course, pH extremes because there are lots of sulfuric acids in there. So you have a very, previously, they were originally classified as extremophiles. Actually, archaeals are known for being extremophiles. So here are some comparison between the bacterial and the archaeal cells. So we have this plasma membrane lipids. So first is the plasma membrane lipids. For the bacteria, it's quite similar to the eukaryotic cells. We have ester-linked phospholipids. But when we look at the archaeal plasma membrane lipids, they are slightly different. Yes, they still have glycerol, but however, instead of glycerophospholipids, they have diethers of glycerol or glycerol tetraethers. Later, we will look more closely at the molecular structure of their membrane lipids. But the differences in the membrane lipids allows them to actually survive in very extreme conditions. And then, the second property that they are different in is the cell wall constituents. So in bacteria, the cell wall constituents, as we have discussed in the previous lecture, they are made of peptidoglycan layers. Although some bacteria, like the Mycobacterium tuberculosis, does not have has a slightly different peptidoglycan wall. Actually, it's made of mycolic acid, but generally they are this peptidoglycan layer. But for the archaea, they have a more diverse cell wall constituents. So the most common is what you call the S layer. Later we will talk about the S layer. There are also some polysaccharides and proteins in this cell wall constituents. While some archaeal bacteria lack cell wall. One thing in common is that archaeals Archaea bacteria do not have peptidoglycans in their cell wall, whereas for the bacteria, we have the peptidoglycans. Now, some commonalities between them, they have inclusion bodies, and that is why sometimes we mistake Archaea under the microscope as, of course, the same, from the same group of organisms. They have the same ribosome size at a 70S ribosome density. And for these chromosomal structures, the bacteria can have circular double-stranded DNA, the same with the archaea. Simple DNA structure. Basically, we have just circular double-stranded DNA. Yes, they both have plasmids, but for the plasmids of the bacteria, some are circular, some are linear, whereas the archaeas have circular double-stranded DNA plasmids. So bacteria, because they tend to be more sensitive to their environment, they tend to form capsules and slime layers as a way to protect themselves. Whereas the archaea, as extremophiles, they have their plasma membrane that is very, very tough, very, very good, um, that provides a good protection. That is why they are rare. Very rarely they form slime layers or capsule, because these capsules or slime layers are a form of protection against the environment. And the archaeas have, although they do live in extreme environments, they have very tough membrane lipids. They compensate it with their membrane lipids. Rather, their cell membrane and cell wall compositions. Now, for the formations of their pili and fimbriae, it's common in bacteria, but for the archaea, more common are the pili and the archaealia. So that's their own version of your pili. And then we have motilities. So how do they move around? So we know of the flagellum, which is a whip-like tail that allows the bacteria to move. For the archaea, they have their own version. We call them the archaeolum. For the cytoskeletal elements, they are homologs of tubulin and actin, the same with archaea. So both have homologous protein homologs of tubulin and actin. Tubulin and actin are the cytoskeletal elements in eukaryotic cells. And yes, both of them produce extracellular vesicles. So as you can see, there are some points that are similar with the bacteria. That is why we originally mistake them for such a group, a single group. But uh, their habitats, their tendencies to live in extreme environments are one of the main reasons why archaea and bacteria, or one of the main differences if we look at their, especially in the cell walls and the plasma membranes. Now look at the shape, arrangement, and size. So the most common shapes and size are the cocci and the rods, but other shapes similar to bacteria can also exist for them. However, no spirochete or mycelial forms of archaea are yet to be discovered, and unique branch and flat shapes have been observed. So the sizes of this coccyx and rod sizes of this archaea, 1 to 2 micrometers wide to 1 to 5 micrometers long for the rods, and the cocci, which are the circular ones, 1 to 3 micrometers in diameter. So because of their forms and shapes, even under the microscope, some of us might even mistake them for bacteria. So the smallest observed archaea is around zero point 0.2 micrometer in diameter, whereas the largest so far observed is a multicellular form of an archaea, which is around 30 millimeters in length, which is quite surprising because they are considered as unicellular, but this one is a multicellular form. So here are some morphological views of them. So this is a cluster of cocci cluster. So we have this branch form, which is quite surprising for a unicellular organism, or rather, bacterias, on the other hand, very rarely, if ever, they form this branch forms, but archaea do form this branch archaeal cell structures. So here are some structures of them. So nanoarchaeote, and with its host. So you have its this one, a nanoarchaeote with its host, and then we have this filamentous archaeon and this bacteria. a bacterial biofilm growing atop your um, a large archaeon of a relatively large archaeal cell. So, as I said before, we have distinctive archaeal envelopes. Their envelopes are quite diverse because it allows them to actually live in very extreme conditions. So in this part, we'll look at archaeal cell envelope and what are the component layers. To compare and contrast archaea versus bacterial cell envelope in terms of their structure, molecular makeup, and functions. And compare and contrast nutrient uptake mechanisms observed in bacteria and archaea. So let's look at the archaeal cell envelopes. So the archaeal cell envelopes differ from bacterial cell envelopes in the molecular makeup and organization. We have what you call an S layer, or rather an S well, we call it an S layer, but they are actually made up of subunits of protein structures. These are arranged outside the plasma membrane. So basically, these are their quote unquote the cell walls. So some bacteria lack, some archaea lack the cell walls, but they compensate it with carbohydrate covering. So slime layers are observed to mediate cell-to-cell interaction, but we do not really know much about it because it's quite rare for archaea. So here is an archaeal lipids. So this is, if you look at this, similar to a normal bilipid structure. So you have this bilipid layer again, a polar head and a non-polar tail. But if you look closely at the tail consists of quite unique lipids. They have hydrocarbons. So when we say hydrocarbons, of course we have carbons and hydrogens, but they are made up from isoprene units. They are branched. Unlike for the bacterials and even for us eukaryotic animal cells and plant cells, our um, our lipids are not branched. We do have kinks, double bonds that produce kinks, but for this one, they actually have branches in their hydrocarbon tails. So some of them are not, in even some of them, rather. So another form of a bilayer, not exactly this one is not exactly a bilayer. So the plasma membrane, another form of plasma membrane for the bacteria, is not a bilayer, but rather a monolayer, but with a longer, uh, with a long tail again, or rather a longer chain of hydrocarbons because it has the similar sizes in length to um, this bilayer structure, but this is just a single cell, not cell, one molecular structure. So one layer, one solid molecular structure with two polar heads on both ends, and then two very, very long tails. And if you look at their tails, they actually have here cyclic hydrocarbons in their monolayer structure. So instead of a bilayer where the two layers are just bound together by the relatively weak van der Waals, but when taken together, it becomes quite strong if there are thousands of them or millions of them. So instead of there's the van der Waals force maintaining this bilayer together, here the monolayer they are actually covalently bonded with one another to form this single layer. Um, I can say this is a bilipid, but it's a single layer structure. We have two polar heads. It actually acts a similar way to the bilayer, but this one is stronger because of the covalent bonds. This is, again, one of the reasons why they can actually survive in very extreme environments. So the archaeal membrane lipids have two major types, a glycerol diether lipids. These are hydrocarbons, take note, 20 carbons, 20 carbon hydrocarbons attached to your glycerol. So C20. These C20s makes your bilayer structures. For this, the other type is the monolayer structure. This is a tetraether lipids. We call it a tetraether because you form ether bonds 1, 2, 3, and 4 because you have two tails connecting two heads. So you have basically four connections there. So you have a four ether connection. So it's a tetraether connections. So they are more rigid. that makes this bilayer, sorry, it's not a bilayer, it's a monolayer. This cell membrane layer stronger and rigid. It resists external forces and external stress. So you have polar phospholipids, sulfolipids, glycolipids, and unique lipids are also found in these archaeal membranes. So we look closely at this lipid membrane structure. So these are the bilayers. So as you can see, this is the bilayer for the bacterial cells, and actually sometimes in some eukaryotic cells. So you have here no branches, so straight chain molecules. Some of them have double bonds, and that causes kinks in their structure that regulates the fluidity of the membrane. On the other hand, you have these archaeal lipids, which actually have doesn't have kinks, but rather it has branches in itself. So this is the bilayer version. You have this monolayer version. As you can see, a single molecule, but a very long tail of 40 carbon tail that are linked on both ends to a polar group on the head. So two polar groups on the head. So this is the chain with isoprene subunits uh, derivatives. So you have branches, and this one also have branches, but in addition to that branches, they do also have this circular cyclopentanes. There's cyclopentanes here that also produces more volume and bulk to this monolayer structure. So this, all in all, this uniqueness produces unique characteristics to the archaeal plasma membrane structure. So this is for the plasma membrane. Let's look at their cell envelope. So some of them have cell wall, some do not actually have a cell wall, but the envelope, uh, basically the structure covering their plasma membrane, the most common for archaea are the S layer. So as I said, this S layer is actually made up of subunits of proteins. So here, the circular ones is the S layer. So here, the first one in the example is a simple S layer, but there are also varieties. So the S layer can have an additional carbohydrate or even a protein sheet above the S layer, or it can have carbohydrates beneath the S layer, in between the S layer and the plasma membrane. Or basically, this one does not have an S layer itself, but rather have its a carbohydrate covering instead of the S layer. Some archaea also have a double plasma membrane structure. So this is the double membrane of this S layer structure. So these are the varieties of the S layer for the archaeal cells. So what exactly is this S layer? So S layer is a protein shell of this for the archaea. So they are made up of protein subunits. So here is an example of this S layer. They are embedded, basically embedded or in the plasma membrane. So they have hydrophobic tails that extend into the cell membrane in the plasma membrane that anchors it here. And then you have this formation of the polar S layer heads. So basically, based on this formation, they are dynamic in the membrane of the cells. They can move around since they are technically not bound or anchored to anything. They are drifting into the sea of plasma membrane lipids. So sometimes they are decorated with carbohydrates to allow for a stabilized rafts or basically to add stability to the layer. So aside from the layers, what other structures do we have? We have external vesicles, so extracellular vesicles, and some of them are also nanotubes. So vesicles composed of the plasma membrane and the surrounding cellular material, or the S layers, have been found associated with your archaea. These are cargo packed within include cytoplasmic contents, proteins, and nucleic acids. Now, there are also some tubules, nanotubes found in the archaeal cell. So here is a nanotube here, shown by the white arrow. So here is a nanotube. These are the extracellular vesicles, just like pimples or warts emanating from the surface of the cell. So these are external vesicles budding out. And then we have these nanotubes associated with this one. So they are, although the exact function is yet to be it's not yet well studied, but they are thought to be important for intracellular gene transfer in thermophiles to protect DNA from denaturing in high temperature. So they serve, especially for this nanotubes, they serve as protection for cellular communications. So how about their nutrient uptake? So archaeal cells use many of the same mechanisms for nutrient uptake exhibited in bacteria, and that is why originally we did mistake them for such. They have passive and active transport. So passive diffusion, passive and facilitated diffusion, and even active transport. So one of the transport systems found in Archaea is the phosphoenol pyruvate sugar phosphotransferase system for the some archaeal cells. Okay, so let's move on to the next part, which is archaeal cytoplasms. So archaeal cytoplasm, the interior of them, the archaeal cell is similar to your bacterial cytoplasm. So let's look at the similarities and difference with the bacteria. So again, they are very similar. They do not have membrane enclosed organelles. Basically, they are technically prokaryotic because they do not have a known nucleus and even membrane-bound organelles. But they do have inclusion bodies. And for the Archaea, these are usually gas vacuoles, meaning they contain air. So the components of the cytoplasm, you find the ribosomes. They do not have a nucleus, but they do have a nucleoid regions and the plasmids. But aside from that, there are some differences also in structure. So let's look at the ribosomes. So we have the same sizes. If when we say similarity, these are in terms of the size, both are a 70S ribosome made up of a 50S and a 30S subunit. However, the exact composition, when we say same size, technically same density overall, but we look at their chemical composition, they are different from the bacterial ribosome. In fact, the archaeal ribosome, in terms of ribosome similarity, they are more similar in terms of composition to the eukaryotic ribosome. The rRNA molecule is similar to size, but the sequence of the nucleotide is not similar. The ribosomal sequence for the archaea are much closer to the eukaryotic ribosome than that of the bacterial in terms of sequence, but in terms of size, they are the same. Rather, they are the same to bacteria in terms of size, but sequence is different. So for the protein composition, the archaeal ribosome have more R proteins. The different composition makes archaeal ribosomes unaffected by antibiotics that target your ribosome. And so, although very rarely are some archaeal pathogenic, but when they are pathogenic, they cannot be killed by normal antibiotics because they do not have, although similar in size, only the size is similar. The chemical properties are different. Hence, normal antibiotics won't work on them. So again, Archaea is much more similar to the eukaryotic ribosome in terms of its biochemical characteristics than the bacterial ribosome. Now let's look at the nucleoids. The nucleoid region is a region in the cytoplasm that contains the circular chromosome and the nucleoid-associated proteins. So both of them are irregularly shaped regions in the bacteria and the archaea, but they are not membrane-bound, with a few exceptions. So we have found some evidences of polyploidy, but again, the challenge in studying archaea is because they are quite fastidious. When we do say fastidious, remember when I said they are extremophiles, they tend to live on extreme conditions, and that is why culturing them in the laboratory under normal conditions is very challenging because they cannot live in normal conditions. They are well adapted to very high temperatures, but not to lower or normal temperatures. That's why they are difficult to grow in the lab. We can only source them from their original habitat, and then when we move them from there, they usually die because they are not adapted to the normal conditions that we live in. Now, as the nucleoid region have also supercoiling and nucleoid-associated proteins. So again, the supercoiling is a mechanism to assist in folding and chromosome condensation so that it becomes well packaged and doesn't tangle inside the cell. So they also have histones that organize their chromosome into nucleosomes that are quite similar the histones. The histones are actually a characteristic of eukaryotes rather than prokaryotes. And we find them in Archaea. Now, for the external structures for attachment and motility, so let's look at the bacterial and archaeal pili and the archaeal flagella, the archaeali. Okay, so let's look at the pili first. A pili is composed of pilin proteins that are made from the cytoplasm and then anchored to a protein complex in the plasma membranes. So we have two archaeal pili, the cannulae, a hollow tube-like structure on the surface of the thermophilic archaea. It is shown that daughter cells that arise from cell division remain connected to each other by this cannulae. And then another archaeal pili is the hami. These hami resemble the grappling hooks, which may function to attach the cells to surfaces. Basically, um, mostly supposition, but anyway, we see them in biofilm communities. So in communities of bacteria, not sorry, archaea. So here are the cannulae and hami. So here is the cannulae. So they are basically strings attaching these two archaeal cells here. And then this is the hami with hooks in the end that allows them to hook to surfaces. Now for the motility. So the flagella of the archaea, we call them archaeali. So they are thinner than the normal flagella of bacteria. So some of these flagella, or rather archaeala, are made of more than one type of protein. So they do not have a hollow filament like the flagella of the bacteria, but they are also moved by rotation. And the rotation is powered by a motor from ATP hydrolysis. So the ATP hydrolysis produces the proton motive force. So the direction moves the cell forwards or backwards rather than the running and tumbling movement seen in some bacteria. So swimming motility for these archaea are quite fast. So that's generally the look we have in archaeal cells. So take home message. The archaeal domain, as I said, is relatively new. Not exactly newly discovered, but we have discovered them since before, but we thought of them as bacteria. That is why it's a relatively new domain because we just now realized they are their own separate group. So it is critical to understand to remember that there are similarities with the bacteria. That is why we confuse them at first as bacteria, but in reality they are different. So um, basically you have this archaeal cell. In fact, when you look at um, the phylogenetic tree using this ribosomal DNA, archaeal bacteria are more similar to eukarya in terms of biochemistry than the actual bacteria. Okay, so that's it for this lecture.